Okay, recording started. Welcome to everybody. I'm very happy to see you all here again for this new webinar. Today, I'm very happy to be here with Marie-Laure Pilet-Nayel. She comes from IRN. She's a researcher that specialized for many years at this point on quantitative genetic, plant breeding, etc. And today we are going to talk again about root rot, which has been, and the general root disease has been a growing theme and topic in our webinars. And today, specifically, we are going to talk about root rot resistance in pea. So please, Marie-Laure, sorry, and welcome, and you have the mic.
Okay, thank you very much for the invitation to present today. I'm going to present you our research work on genetic control of Aphanomyces root rot resistance in pea. Pea has been the most cultivated grain legume crop in France for more than 30 years, but growing areas have dramatically declined since the mid-90s, especially due to yield instability of the crop generated by Aphanomyces root rot disease, which has been the most damaging root disease for 25 years in France. Aphanomyces root rot is caused by a soil-borne oomycete pathogen, Aphanomyces euteiches, which oospores resulting from sexual reproduction produced by flagellate zoospores, which are the infectious unit of the pathogen. The symptoms of the disease are root browning at early plant stages, and at a later stage, yellowing and dwarfism on the aerial parts of the plant. The pathogen has a broad host spectrum in legumes, and two main pathotypes have been identified on pea, pathotype 1 and 3, using a differential set of pea genotypes. Disease management methods include the avoidance of infested fields, rotations with non-host or resistant legumes, such as faba bean, and the choice of also sowing date with winter peas, which have shown to partially escape the disease. And also tolerant varieties are now available in France for about 5 years, but still requiring more progress in pea breeding to increase resistance levels. Pea breeding for resistance to Aphanomyces root rot has been very challenging for several decades worldwide due to the polygenic inheritance of resistance, partial level of resistance, but also linkages with undesirable traits. And few sources of resistance identified in the Pisum sativum species and its few related species which are intercrossable with pea, and also the difficulty of breeding for polygenic agronomic traits as yield at the same time. So therefore, breeding strategies for high and durable levels of resistance are more complex than for resistance controlled by single or a few genes, and will require to diversify, finally select and combine genetic loci associated with the partial resistance. So to diversify resistance genetic loci, we conducted in the past years a large screening program of about 1,700 Pisum accessions for resistance to Aphanomyces euteiches in controlled conditions. This resulted in the selection of 20 Pisum accessions showing partial resistance, which was validated in the field. And among these accessions, 4 main sources of resistance were used for genetic analysis of partial resistance. These 4 lines are 902131, 902079, PI 18693, and 552. And these sources of resistance were used as parental lines of recombinant E-brain line populations and as sources of resistance in a French private recurrent phenotypic breeding program. And this program resulted in the selection of breeding lines of agronomic type with higher levels of partial resistance. These lines were a major part of the P. Aphanomyces collection, which was used as for GWAS analysis of resistance. And also two other sources of resistance, E11, which is a wild Egyptian accession, and Lysa, which is a fodder German cultivar, was also used for genetic analysis in advanced backrust populations. From these sources of resistance, we conducted extensive genetic analysis of Aphanomyces resistance, and we published recently an overview of the diversity of QTL detected by integrating QTL mapping studies in the advanced backrust recombinant inbred lines populations and GWAS studies in the P. Aphanomyces collection, using SNP common to SNP genotyping data common to between the different types of populations. And we identified 10 genetic regions which were consistently detected, distributed over all the P chromosomes, but one including especially, yes, just 6 chromosomes. And especially, we detected 2 major effect QTL. The first one on AE named AEPS 7.6, located on chromosome 7 and efficient against Aphanomyces euteiches isolates from pathotype 1. And the second one on chromosome 4, namely AEPS 4.5, which is efficient against isolates from pathotype 3. Negative linkages were also observed depending on the sources of resistance alleles in different QTL regions between resistance and morphological or developmental traits, including normal leaves at AEPS 1.2, anthocyanin production and late flowering at AEPS 2.2, and late flowering and resistance at AEPS 3.1, and yield at the major effect QTL AEPS 7.6. We focused on the major effect QTLs to perform fine mapping of their confidence interval in order to try to identify candidate causal polymorphism and diagnostic markers for resistance. First, we fine mapped the QTL AEPS 4.5, which we redetected with the major effect from the Puget 90-20-79 recombinant inbred line populations for resistance to a reference isolate for pathotype 3, AE109. We screened the 4 recombinant plants in the QTL interval from progenies derived from heterozygous near isogenic lines. And by marker phenotype association analysis on 15 recombinants, we reduced the QTL to a 3 megabase interval on the version 1A of the P reference genome. And this interval contained 50 annotated genes, among which 3 putative candidate genes encoding dead box helicase, C2 protein, and a subtilase family protein also. And especially the second gene was one of the genes identified by bulk segment RNA-seq in a previous Canadian study in the QTL interval. That's okay. Second, we fine mapped the QTL AEPS 7.6 that we especially detected with a major effect from the DSP 90-21-31 real population for resistance to our French reference isolate for pathotype 1 RB84, and using 40 recombinants identified from the screening of more than 6,000 plants from large near isogenic like progenies. We reduced the interval to a 1.9 megabase interval, and by GWAS analysis, we in a P architecture and multi-stress collection, which is another collection which was sequenced in the exome parts of the genome, we identified a major QTL peak exactly in the same interval, in the 1.9 megabase interval. And we identified in this interval 33 genes on the version 2 of the P reference genome. We also conducted RNA seq analysis between NILs with whose genomes differed only by a large introgression in the QTR region. And the results specifically identify 9 constitutive differentially expressed genes between NILs with or without the QTL, but none of these genes were identified in the fine mapped interval, and structural variants were also identified from RNA sequences in genes in the fine mapped interval, and also in genes belonging to an NBS-LRR cluster located near the fine mapped interval. And functional validations of candidate genes is ongoing by Tilling, and it's also planned by CRISPR-Cas9. So to investigate the effect of combining QTL on increasing the level of resistance, we created near isogenic lines carrying resistance alleles at 0, 1, 2, or 3 main QTLs by marker-assisted backcrossing into agronomic lines. The NILs were evaluated for resistance in growth chamber, in greenhouse, and in multi-local and multi-year field infested nurseries or yield-infested versus healthy plots, using the INRAE French breeders Aphanomyces field network. Several QTL combinations were confirmed for their increased effect on resistance, especially when including the major effect QTL AEPS 7.6 alone or in combination with minor effect QTL, especially the AEPS 5.1 or 4.1, and some combinations of minor effect QTLs also showed increased resistance in yields, but also sometimes with undesired linkages with especially late flowering or colored flowers. More recently, we pyramided NILs, we pyramided QTLs by intercrossing best NILs, each carrying 2 or 3 QTLs with resistance alleles originating from different sources of resistance. And this resulted in the production and selection of more than 200 NILs with new combinations of 2 to 5 QTLs. We evaluated the impact of the disease on the vegetative and reproductive plant development on series of NILs in inoculated versus non-inoculated conditions using a test developed in greenhouse. We showed an effect on QTL pyramiding on reducing the impact of the disease on aerial parts of the plant. And we also observed an effect of QTL pyramiding on reducing oospore production in the roots using a protocol of oospore extraction from the roots before countings. And here on the figure, we can see that NILs combining 4 or 5 QTLs had a significantly lower number of oospores in their roots than parental NILs carrying 2 or 3 QTLs, and then the susceptible control lines. And also, it was not, although it was not as low as in the resistant control here. So, however, we also observed that this QTL pyramiding effect depended on the genetic background. And in addition, allelic linkages were observed between resistance and undesired traits due to large introgression sizes at the QTL regions in the NILs. These results therefore led us to move towards the idea that genomic selection is in large breeding populations from multiple sources of resistance would be a new type of scheme more suitable to accumulating minor effect QTLs and breaking negative linkages. So in order to develop this type of new breeding scheme, we have created a large multi-parent advanced generation intercross population, or efficiently pyramiding resistance and agronomic alleles. And the population comprised of about 1,800 lines and was fixed at the major effect QTL AEPS 7.6 from the early generations. And we recently phenotyped this population for resistance to Aphanomyces root rot in controlled conditions. And we showed a variation for resistance with some lines having higher levels of resistance than the best resistant control lines. The population will be genotyped using the multi-grain legume species s3e07 chip under development in the European BELIS project, which will include QTL-associated SNPs, especially QTL for resistance to Aphanomyces root rot, and whole genome diversity SNPs. And genome-wide marker trait associations and genomic prediction analysis will be performed to identify markers to accelerate the selection and pyramiding of minor effect alleles for Aphanomyces root rot resistance in future breeding schemes. Okay, just to conclude, I would like to say that our research work showed that the quantitative inheritance of resistance in P is composed of major effect and minor effect QTL. Identified genes underlying major effect QTL will be useful for breeding and science. And combining and diversifying resistance QTL is promising to increase levels of P partial resistance. And progress is still required to durably increase the level of resistance of agronomic varieties. And of course, new genomic assisted breeding schemes will be relevant to accumulate both resistance and yield alleles with minor effects. Okay, finally, I just would like to thank all our colleagues from INRAE, Terre Innovia, from Rennes, INRAE, Dijon, Versailles, Mons, French breeders, and colleagues from USDA and our funders for their great contributions to the work presented. And a special thanks to Clément Labot and Théo Le Prévost, who were really major contributors to this work presented today. Thank you very much for your attention. I guess I have to stop the screen.
Thank you, Marie-Laure.
Sorry, I think I have to stop the screen, otherwise I cannot see the audience. Should I again share my screen if you want, or just maybe
fine like that. So thank you. You were pretty fast, but you presented very promising results. So as usual, we have some time for the questions, and I'll also take a look at the chat. Meanwhile, while people is waiting to chat, I remember that we have a feedback form that I linked in the chat. So if you could just write, it's not obviously a judgment on Marie-Laure, but it's a feedback for us to have more and more refined offer for the webinars, and also if you have some themes that you would like to us to explore more for next season of our webinars that will start in September. So we have I see Tom has raised his hand.
Yeah, thank you very much, Marie-Laure. Very interesting presentation and excellent progress. Could you just say a little bit more about pathotype 1 and 3 and how you maybe how they differ and how the QTLs, how you feel the QTLs might perform with respect to those different pathotypes? Which of those two is more important in the field in France?
Yes, those pathotypes 1 and 3, initially in the years 2000, Emmanuel Vicker found several pathotypes, 11, but finally only 1 and 3 were the major pathotypes. He found them based on the differential reaction of 6 P genotypes and of isolates, and especially the for pathotype 3, the P genotype MN313 is resistant, and MN313 is not resistant to pathotype 1. So this P genotype is the major genotype which makes the difference between the two pathotypes. And especially we found in France, and we isolated now, I guess, a lot of isolates in France, and we found only isolates from pathotype 1. I mean, MN313 is susceptible to these isolates, but PI 18693 is partially resistant. And at the difference here, people genotypes, and especially 902079 is resistant to pathotype 3 isolates. So, but these kinds of pathotypes were not found in France. And I guess that these types of, we found that these pathotypes were found in some regions in the USA, and I guess in Canada also a little, I guess. But it's, yeah. So this QTL finally, it's expected not to be efficient in France, but it's interesting to know what which genes are analyzed this QTL. But we have a major interest, major focus in France on this major effect QTL, which is not enough anyway for to increase the level of resistance. I hope it was clear.
Yeah, thank you very much.
Okay, see somebody is writing in the chat. I read this message for you. Thank you for a very interesting presentation. Sure. Is there anything known about potential functions of genes in these QTLs?
Yes, for the moment, we don't know the functions of genes. So we have identified, we have a focus on this of the cloning of this QTL, AEPS 7.6. And we have identified candidate genes in the fine mapped intervals. But we still do not communicate on these genes because it's not, we would like to try to see if we can validate them by tilling. But it's very complicated to know if the causal genes could be the NBLR genes close to the interval. If in the fine map interval we have a sequence in introns sequence which could interact with the NBLR genes, it could be a possibility. So we have to sequence also. We have to obtain more sequences of the in this region in the resistant genotypes, especially PI 18693, which is part of the pangenome genotypes sequenced. So more information will be provided by sequences in resistant genotypes, because the candidate genes we found up to now is are located or in Cameo, in the reference P genome Cameo, which is susceptible to Aphanomyces root rot. So it's complicated to tell today which function underlies this QTL. We need more sequences, whole genome sequences in the resistant genotypes, and more validations on the genes on genes.
Thank you, Marie-Laure. Are there other questions? Yes, I see Sabine, a couple of raised hands. One is Sabine Baniza, so you can unmute and talk.
Good morning. Very nice talk, Marie-Laure. I want to follow up on what Tom was asking. I know that Christophe Lemay has been doing field research looking at the evolution of the pathogen in plots where they grew these lines with QTLs. Can you comment a little bit? I assume you're probably familiar with that and involved in that because you must be using your germplasm. What are the dynamics of the pathogen population in response to deploying these resistance QTLs?
Thank you very much, Sabine, for this question. It's a major concern for us in research in France because, and we have developed a field essay, an experimental field essay for 8 years in France in a contaminated field, in which we grew Nils with the major QTL for 8 years in single crop rotations. And we are in the process to characterize the isolates after 8 years of growing of these Nils to see if the aggressiveness of these of the population of isolated has increased. So we still do not have results, but it's a strong hypothesis. We had first results after 4 years of single crop, and we observed a slight higher frequency of more aggressive isolates, but we need to confirm this. And it's really a hypothesis to a strong concern for us if breeders use only this major QTL. First, the level of partial resistance conferred by the QTL will not be enough. So, but moreover, we have a risk that the pathogen population could be filtered. The highest the isolates, which will be the most aggressive, could be selected by these varieties, and then that the population, the Aphanomyces euteiches populations could evolve towards more aggressive, more aggressivity. So it's just necessary in the future varieties to have varieties very resistant. Just no choice because of the evolutive gap. The population, the pathogen population could more easily evolve if we breed tolerant varieties and then after more resistant varieties. It's really indispensable to obtain in varieties high levels of resistance. Yes, I know it's a big challenge, but no choice, I guess, if we want to have a durable resistance. So
thanks.
Thank you.
Thanks. There's a couple other people raising their hands. I see Dae-Kyun Ro. So you can speak.
Hello, thank you. I enjoyed your talk.
Hello.
I'm relatively new to this field, and my question may be too simple. But you indicated fava bean is resistant to the Aphanomyces. And then there are other the legume species like lupins, and others are resistant to Aphanomyces. So do we know the nature of the like faba bean or lupins resistance against Aphanomyces, and perhaps you can learn something from there?
It's a good question. Maybe as faba bean and lupin are resistant, finally we did not focus research efforts on genetic analysis in these species. We have recently conducted genetic analysis of resistance in faba bean. So we have a paper just submitted, and we found some QTLs, but still need to be confirmed, and it's no major genes. And these QTLs in faba bean do not seem to be syntenic to QTLs found in pea. So it's not very well known up to date to the genetic determinism in faba bean. We have a hypothesis also about the effect of these species in the early steps of the pathogens. I mean, on the attraction of zoospores. We have a PhD project currently, who is interested in trying to look at the effect of the root exudates from faba bean and from pea, Niels, towards attractivity of zoospores. And because some papers show that root exudates from faba bean have a repulsive effect on zoospores. So it's interesting to know, to have more research about why faba bean and lupin are resistant or non-host, but we still do not have a lot of knowledge on this. Okay. Thank you.
Thanks.
Okay, I see then probably Lars is there.
Thanks for a very lovely talk, Marie.
Thank you.
My question was more around the development of the population, how you're going to genotype the magic population. You mentioned the belly SNP chip. Can you elaborate a little bit more what the approach is there and which species are going to be captured on that SNP chip?
Yes, so the SNP chip is under development with our colleagues of INRAE of Dijon. We will have 5 species on the chip: pea, faba bean, lupin, lentil, and chickpea. And so the colleagues, Grégoire Robert is in charge of this development with my colleague Clément Lavaux. They have already discussed with the s3e07 core in Australia, because there is a chip also developed gathering several grain legumes, so that we could have bridges between chips, common markers, and so that we could compare results with these chips. But the objective of this chip is that it could be very low cost chip with about 60,000 SNPs, with about 20,000 SNPs for pea, 15 for faba bean, 10 for each of the 3 other species. So it's this chip will gather markers already published in other and especially markers which could have been observed as linked to QTLs. So it would be a very, very useful tool, I guess, for the community, and I think with a low cost.
Thank you. No, that's very nice and clear, and good to see the synergy and a small degree of overlap with the chip that has been developed here in Australia. So
yes,
thank you.
Okay, there were many questions. That's a testament to how interesting was your contribution, Marie-Laure. I don't see anybody else raising their hands or writing in the chat. So I guess this marks the end of our webinar for this month. Thank you to everybody. The webinar will go on our YouTube channel, on our website, and we also have a Blue Sky profile that we recently opened if you are into that. Otherwise, see you all in a month. Bye.
Thank you very much to everyone. Thank you. Thank you very much.